Good morning. My name is Dr.S.Radha, Assistant Professor of SFR College of Department of Microbiology. So I am going to take a topic of forensic science. So under this we are going to see first, that is DNA extraction method. I am going to discuss the basic principle of DNA extraction and how the protocol is performed, then where and all it is applied. So DNA extraction principle first we have to discuss. So this is done with the phenol chloroform extraction method, where we use the two phase of liquid. Usually we are adding to the DNA sample the phenol and chloroform mix of 1:1 ratio, and this is based upon the principle that phenol will remove the hydrophobic interaction of protein and it will precipitate them in the interphase. The lipid will dissolve in the organic phase and the DNA will suspend in the aqueous phase. So for doing this DNA isolation, various source sample we can take. Usually in forensic science, we will take the animal tissue as a sample. So animal tissue could be a buccal swab, hair sample, urine sample or blood sample. Other than that, we can also use the serum sample. The processing of the sample will vary depend upon the source of the sample what we are using for DNA extraction. The method of DNA extraction involves first the sample has to be dissolved in a suitable buffer. In this diagram I have shown that is a swab sample that is dissolved in a buffer. So this buffer what we are using is a tris buffer of pH 8. This will be followed by addition of phenol chloroform mix in a 1:1 ratio, and this has to be vortexed in a rapid way. This will make the cell to break and the content will be released outside, and it will be separated into two phases. In the upper phase, the aqueous solution DNA will be suspended, whereas in the interphase the protein will be there and lipid will be dissolved in the down. We will be transferring the upper aqueous phase to a clean another centrifuge tube, where we will precipitate this by adding ethanol or butanol. After adding the ethanol or butanol, the precipitation process will be followed by a speed centrifuge process, by which DNA will be precipitated and it will form a pellet. And this will be followed by a washing step that will be done with a 70% ethanol. Washing usually it is done by twice. And finally the DNA will be pelleted and it will be suspended in a teeny buffer and it will be stored in -20 degrees Celsius till we use for next experiment. So the next experiment use the observation of DNA that is done using agrogel electrophoresis method. So this apparatus that is used for DNA observation or analysis, here I have shown the diagram where the tank will be filled with a buffer. The boat will be casted with agarose. The agarose will be 0.8% to 1%, and the comb is used for casting the well. And the sample has to be prepared, that is dissolved in tea buffer, will be mixed with the sample loading dye and it will be added to the well. And we have to supply a power, that is it is under the electrophoresis, that is the mobility under electric field. So it will move DNA as it is a negative charge, it will move from cathode to anode, that is from negative to positive. And after doing this gel electrophoresis, we can observe this by using UV transformation technique. So UV translator is an instrument which will provide the UV under which we will placing the gel, and this ethidium bromide what we are using for staining the gel, it will intercalate with the DNA molecule. And when you are observing under the gel, it will illuminate or it will orangish band it will show, by which we can confirm the DNA analysis and what is the prerequisite for all the forensic science experiment what we are doing. And what are the experiment for which the DNA analysis is done is for suspect. If you are finding a suspect in the crime scene, their DNA has to be extracted and it has to be performed on DNA analysis. Other than that, the accused, that is confirmation of accused or to confirm the suspect, and for that is solving the paternity and family relationship, for all the that is crime scene or all the purpose, we are using the DNA extraction as a prerequisite experiment, That has to be done carefully, then only the result can be, that is confirmed for forensic science. Thank you.